Hi, I'm Mr. Baker. We're Unit 3 cells, and we're looking at 3.3, the ways that molecules move across membranes. You'll need to know about a concentration gradient, which is simply you've got more of something in one place than another. And you can think of it as population density for molecules. Molecules will spread out due to the fact that they're in constant motion, and they will move down the concentration gradient. They do this for free, there's no energy used, and the cellular energy molecule is ATP, so no ATP is being used. Cells need to move molecules and things down and also against the gradient. Down, you're moving more things to less things, like food coloring and water. You think of it as moving with the current. If you're going against the gradient, you're moving less things to more things, like moving food into a cell, and you're going against the current. Cells have two options, passive or active transport. Passive, they move out due to diffusion or in. No energy is being moved, and you're moving down the concentration gradient. Active transport, you're going to actively move things, and you're going to use energy in the form of ATP, and this goes against the concentration gradient. But first, passive transport. You have two major options, diffusion and osmosis. Diffusion, no energy is needed. It's going to be dissolved in water, and you've got a drop of food coloring in water showing you how things are diffusing. Diffusing, molecules tend to move down their concentration gradient or spread out. That is simple diffusion. They move on their own without any help. There is also facilitated diffusion, which is helped. Think of a gate being put in that allows the molecules to get in and out. For example, if you're a large molecule, too big, or maybe you're charged like an ion and you need some help. Here's a picture of the gate. It's the purple dude on the right side letting the green things through. Osmosis is the diffusion of water across the membrane. There's a picture of things we've discussed previously, and that brings back into play psi. Psi is a measure of water potential, and you need to know the three rules of psi. The psi of pure water is 0. The more stuff in a solution you have, that means the psi is more negative, and water will always move to where the psi is more negative. Here's a table, and this will help you memorize what's going on and where water moves, and an example, and the actions, and some fancy words like lysis and crinates. The direction osmosis is determined by the previous table, so we'll move quickly. Here is passive transport and osmosis. On the left, you're putting a red blood cell into pure water. Isotonic solution is a red blood cell in like saline water. And hypertonic solution is putting a red blood cell in salt water. Your cell shrinks. That's why you don't drink seawater. So here's a nice table summarizing our passive. You have three options, diffusions, osmosis, and a facilitated diffusion, all moving down the concentration gradient. None of them use energy, and specifics. Here's another GIF on passive transport, which I'll let you watch. Facilitated diffusion on the right has a gate. And in contrast, here is active transport, which is like a gate that burns ATP. Molecules can't move out spontaneously. That's when you're going to have to use, and you're going to have to use a gate or a pump. One example is H plus against the gradient to help you digest food in the stomach. That is an example of active transport. Here's their picture. And then you've got things called proton pumps, and there's a GIF of what's going on. And it's taking protons, which are H pluses, and it spins around and pushes them through from where there is less of them to where there is more, the definition of active transport. There are two other main types of active transport, endocytosis and exocytosis. Endo, inside, moving things into the cell. Exocytosis, moving things out of the cell. You're going to do this for things that are too big. That is a .gif of, looks like immune cells taking in bacteria and eventually chewing them up. Here is a table showing you your active transport with your pumps, your endocytosis, and your exocytosis. You're moving always against the gradient. You're always burning ATP. Pumps are motorized gates. Active, or the endocytosis, is moving stuff into the cell, and there are three specific types: pinocytosis, phagocytosis, and receptor mediated. More about that. And exo is to exit, moving out of the cell. So phagocytosis is cell eating. Phago is the Greek for eating. Pinocytosis is cell drinking, and surprisingly, pino is Greek for drink, and then there's something called receptor-mediated endocytosis. So here is your phagocytosis and recycling the bacteria eating, or the bacteria getting eaten. Receptor-mediated endocytosis, one example is LDL, which is low density lipoprotein, that's cholesterol. You clear cholesterol, you have a receptor, the LDL binds to it, your cell pulls it in, and it chews it up. People that don't have this receptor will have increases in cholesterol, which can lead to buildup of cholesterol in the bloodstream. Here's some pictures. And a buildup of cholesterol in the bloodstream will end up in the heart, clogging hearts, and causing familial hypercholesteremia. And here's the end result of that. Healthy artery partially blocked and one that's completely blocked. Those can lead to heart attacks. The other interesting things about this receptor-mediated endocytosis, it's how some viruses gain entry into the cells, including SARS-CoV-2. SARS-CoV-2 binds to ACE2 inhibitor, which once it binds to it, it allows it to gain entry into the cell and do what viruses do, which is make more of themselves. Here's the table one more time with both passive and active. Memorize that. Here is a couple review.gifs for you. And that's what you'll need to know for your next quiz. Study hard.